Symposium. Today again we have 3 sessions. So to start with the session, the first session is on translation and post-translation modification. That is chaired by Professor Marsha Roshner. Just to give a brief introduction about her, she is Charles Huggins Professor at Ben May Department for Cancer Research, University of Chicago, USA. So now I welcome our chair, Professor Marsha, to start with the session, and please welcome our keynote speaker.
Okay, thank you very much. Our first speaker actually needs no introduction. He's Dieter Sohl. He was one of the instrumental people who worked with Gobin, doing work that led to the genetic code, understanding the genetic code. He tells me that after he left Gobin's lab in '67, he went to Yale, and he stayed there ever since. And I know for a fact that most of his publications are in some of the very top journals, but I think what I was stunned to hear recently is that he has written over 600 publications. So maybe I should quit now. So he's a pioneer in synthetic biology, and Dieter's going to talk about the genetic code in the age of synthetic biology. Dieter.
Thank you, Mark. Good morning. It is really a pleasure to be here, to be in India again, to be invited by all the people I know well, and have a reunion of all the people that have made me what I became. I have to thank Govind for addicting me to the energetic pursuit of top science. And all the people who were in the lab at that time, and you can see here, this is still a picture from Madison. The code was solved. You see we were very young. Even Echo looks a little younger than she is today. And this was going off to the Federation meeting in 1966 when all the implications of the code were discussed, etc. So it was really a lovely time that we had there. I really would like to go through the genetic code as I see it today. And I will give you a very fast run through the code as I see it coming out from publications that we had in the last few years. So the code in 65, I don't need to give you any introduction, but the code is not just a table and a lifeless document. It gets implicated all the time and redefined all the time. So the tRNA So the tRNA decodes with the help of the genetic message, with the help of amino acid tRNA synthetases. And here, the amino acid tRNA synthetase, let's say prolyl tRNA synthetase, takes proline from the amino acid here and pairs it with the tRNA, with the anticodon for proline, and prolyl tRNA, pro-tRNA pro. This proline is proline attached to tRNA that designates the anticodon, will then be put in a ribosome and will put proline into a protein. So you know all this, but what I would like to state, the genetic code and transfer RNA evolved together. The amino acid tRNA synthetase, on the other hand, that are essential for decoding the code, did not evolve at the same time. Very likely there might have been RNA enzymes that did it, and there will actually be a number. There are 3 examples that we found that are still, where RNA enzymes are still involved in making amino acid tRNA, and I may or may not be able to get to this. The code is not always accurate. So for instance, cysteine can also be attached by prolyl tRNA synthetase to proline, and this exists. But a paper that is largely forgotten shows that already in 1962, in a beautiful review, Richmond showed that there are many amino acid analogs that can be incorporated into proteins during normal protein synthesis. So you don't need to change anything. You don't have to have different synthetases, different tRNAs, they just go in. And this is something that the field forgot for a significant number of years, at least 30 years. So the amino acid diversity that you see here, and I usually ask the question I ask here, how many amino acids do you think exist in normal proteins? Sorry? 20. It's known that since the 70s, by the studies in Babram, Cambridge, in plant sciences, that about 300 amino acids found in normal proteins, and there are recent Chinese publications that claim that you find at least 1,100. Whether this is true or not, I do not know. So in any case, this is simply some of the amino acids we are going to discuss very briefly. Cysteine. We discussed pyrrolysine, that will put these analogs on the tRNA, but this is the amino acid diversity. So I would like to give you 3 slides on what we know about amino acid tRNA synthetases, and I have to say, I admit that I didn't realize until 15 years ago, 10 years ago, that the synthetases are not what I thought they might be. So the synthetases discriminate well against all the other canonical amino acids, the 20 amino acids that you said. And they have perfect discrimination mechanisms against anything in bacterial proteome or any organismal metabolome. However, the amino acids should be polyspecific, i.e. charging several or many non-canonical amino acids, and I will show you some structures of those. And we really were all blinkered and felt that the synthetases will exclude everything. But why would a synthetase evolve, or E. coli or an organism evolve an amino acid synthetase to discriminate some chemical structure that it has never met before, and it may never meet in the next million years. So this has to be like this. So I simply show you 2 slides that we showed that the substrate range of wild type E. coli Trip-RS has, I don't have time to go through the experimental assay, it's a read-through of superfolder protein. All these amino acids here that you see here are activated to some extent by wild-type tryptophenyl tRNA synthetase. And if you do the same with tyrosine tRNA synthetase, you get a similar reaction. And these, for instance, such as PULAR2, are activated both by tyrosine and by tryptophenyl tRNA synthetase. So this simply shows that the amino acid tRNA synthetase are polyspecific and charge several or many non-canonical tRNA, non-canonical amino acids, and I come back to this. And this is work done by Eric and by Chengguang. So, what do we know about today's genetic code? Crick in 1968 assumed that the code is a frozen accident incapable of further evolution. The question really was, how did the genetic code evolve? And it's very clear, if the code evolves, it has to go via mutations in codons. Mutations in codons, of course, immediately make mutant proteins, which means it destabilized the proteome in modern language. And it was believed that the proteome has to be absolutely accurate for organisms to survive. So this is why Crick had this view. Today we know that the known alterations, there are 16 codons where you actually see in blue there are different meanings for those codons in different organisms. And furthermore, there are two additional amino acids, selenocysteine, encoded by UGA, and pyrrolysine, encoded by UAG. So nature already told us how one would go ahead and extend the genetic code by recoding stop codons. And there are two ways that scientists have continued this, by reassigning UGA or UAG stop codons, or by recoding of sense codons. To give you a quick example here, if you look for codon reassignment, then in organelles in mitochondria, all the amino acids that you see here in red are simply derived from what you see here in black or blue. The take-home message is it's actually very easy to do so, and I'll give you an example here where we showed what is actually happening. It was known already for the last 40 years that yeast mitochondria are missing 4 leucine codons. The Cu leucine codons are missing, and so what you see in Candida albicans, there's leucyl tRNA synthetase, and this leucine, so these 4 codons are there, and there's a duplication of his RS, and this is what you see. If you go from albicans to lactis, then you see that this tRNA has been deleted, so only these leucine codons now exist in the mitochondrion. But if you then go to cerevisiae, then one of the histidine tRNAs is actually changing here and can be read by threonine tRNA synthetase. So this histone-derived threonine tRNA now uses the codon CUN that you also see we're missing there. And should you want to go to Ashby gossipy, then you have alanine instead of this. So it is very easy by just a simple base change or base changes in transfer RNAs to change the code. This is an example that we've had more recently. SR1 is an uncultivated member of the oral microbiome. And we actually got the sequence from single genome, single cell sequence, but 85% of the genes with in-frame UGA codons were mostly glycine positions in other organisms. So the take-home message is there's a new transfer RNA, which is has the anticodon here is UGA, and it's a glycine tRNA. And as you can see, it is second most important abundant codon in the organism. So this glycine code, this organism now has 5 glycine codons, and not just 4. And I illustrate with this one a consequence of it. And the consequence is that you know all about horizontal gene transfer. But horizontal gene transfer is when you accept the DNA from another organism. But it has to be a lingua franca that the organism and I will actually have the same code. Otherwise, it's not of use to me. So the fact is, if you do such a recoding experiment, it means that the organism is protected in a way from horizontal gene transfer because no one else can use it. So this is one way of isolating organisms as far as translation is concerned. You probably know that transfer RNAs, especially mammalian transfer RNAs, have very many, let's say 30% of the transfer RNAs are modified, bases are modified. And one of the major base positions is in position 37. That's the one position after the anticodon and is before the anticodon. And you can just see that you have a really large variety. I would like to show you just how complex it is to enforce the genetic code. So if you have oxygen here, that uridine, then of course you can see that it recognizes A ending codons and G ending codons exactly the same fashion. But should this be transformed to thiouridine or possibly to selenouridine, then it is different. Then you can actually see that there is far more A recognized than G recognized. And this is actually the basis why you can then get down to A ending codon recognition, distinct from G ending codon recognition. And it's actually the set of codon-anticodon interactions that was missing in the wobble hypothesis. So you can actually change the code by this. And simply to show you how more complex it is, the enzyme, for instance, 2-thiouridine synthase or 4-thiouridine synthase, or this is an enzyme in cysteine metabolism, they actually require iron sulfur clusters. And these are unusual iron sulfur clusters, and they are essential for enzyme activity. So if you would like to really have the code properly expressed, you have to see that even the iron sulfur clusters are properly inserted. I simply want to show you how many different instances come together for maintaining the genetic code. Something else we saw, this is carbon source dependent genetic code expansion. Acidoholobium arabaticum is a bacterium that, depending on the carbon source you grow it, if this grows on pyruvate, then the proteome has 20 amino acids, absolutely everything is fine. However, if you grow it on trimethylamine, pyrrolysine is being made, and then the proteome expands to 21 amino acids. There's absolutely no problem. You can use the organism after it grew in trimethylamine, put it back in pyruvate, the proteome has 20 amino acids. So you see a phenomenal flexibility that the organism does not necessarily die if you have taken an amino acid away. And so we went ahead and tried to do the following. Ilka Heinemann and Patrick O'Donoghue, who are assistant professors in London, Ontario, decided we should try this. And the tRNA, pile T, is of course the tRNA for pyrrolysine. So we simply excised this and deleted it. And when you did this, you could see that the deletion will no longer grow on trimethylamine, because pyrrolysine is absent. But it still grows on methanol. So we then said, fine, why don't we go ahead and try a proteomic analysis to see what happens? And this is the final result. There are several ways, of course, that you know by which methanogenesis occurs. So methanogenesis for methanol is really very important, and it's very good, and it's very easy. And this we have in our organism with 21 amino acids, while we are now down here. This is down-regulated when we have our 20 amino acids, this is upregulated. So this methanogenic pathway you find with 20 amino acids, this you find with 21, and there is really then a difference in the carbon requirement and metabolism. So why do the synthetic biologists dream about the genetic code? And based on some of the early work by Tom Rajmandari, it's believed that 32 codons are sufficient to encode all the genetic information in genomes. So you can now simply go ahead and re-synthesize E. coli or other genomes and decide how many codons you could leave out by replacing multiple synonymous codons with one other one. And this should leave 29 codons for the insertion of additional amino acids. 61 codons are used for amino acids, so minus 32 should be this. But there are great challenges, because to rewire translation by making orthogonal translation systems, you have to re-engineer the transfer RNA, the amino acid tRNA synthetases, and the elongation factors. And this, the cell, of course, would like to prevent under all conditions, because you touch the protein quality control. So reassigning UAG to stop to a non-canonical amino acid is these days done mainly by pyrolysyl tRNA synthetase. So pyrolysyl tRNA synthetase puts pyrrolysine on the tRNA, pyrrolysine tRNA that recognizes UAG, and then you can make a pile-containing product. And this is what has been done over the last 10 years in many instances, and I'll give you examples. So you have an original orthogonal amino acid tRNA synthetase pair, and this pair, for instance, pyrrolysyl tRNA synthetase tRNA pile, should not interact with the other amino acid tRNA synthetase of the organism, nor the tRNA should be charged by any of the synthetases of the organism. So this is orthogonality. And pylorez, we now know, is the ideal synthetase for genetic code expansion. So the genetic code can be expanded with pylorez. This is just a small example of the over 110 non-canonical amino acids that can currently be charged with pyrrolysyl tRNA synthetase. And some individual enzymes, just an individual enzyme, will actually charge more than 20 of these non-canonical amino acids and insert it into protein. So I'm referring to something that was just published in a December issue of Nature Chemical Biology. Pyrrolysyl tRNA synthetase has two domains, a C-terminal domain and N-terminal domain in archaea. The N-terminal domain is exceedingly insoluble, and therefore we only have structures of the C-terminal domain with and without tRNA. I'll show it in a minute. And we showed already long ago that the C-terminal domain is active only in vivo in the presence of the N-terminal domain. So this then gave rise to the following. So we had a paper in Nature to show the co-crystal structure of a C-terminal domain of the pylorez with the tRNA, and unlike other amino acid synthetase tRNA complexes, this is the enzyme that reaches into the inside of the L shape, and it has a very intricate specialized synthetase tRNA interaction interface, and that I will exemplify in a minute. So we went ahead to look for molecular evolution tools of pyrrolysyl tRNA synthetase with PACE and PANZER. PACE is phage-assisted continuous evolution developed by David Liu at Harvard 6 years ago, which is very simple but very difficult to set up and requires complex equipment. And we decided to make a poor man's method out of it, phage-assisted non-continuous evolution. The methods are based on the phage M13 replication, so this is of course very, very much faster than E. coli. And applications of these methods, and this is the good thing about PACE and PANZER, which is different from MAGE which is being used by many of the synthetic biologists and genome engineers to do genomes, does not require any prior structural knowledge. Otherwise, you need to know you would like to mutationize this because this happens to be the active site or half the active site or whatever. So you can do this. And in this particular case, we showed that both methods give very similar results, and we have isolation of pylorus variants up to 45-fold increased catalytic activity, and mutations are located in an N-terminal domain. And just, I won't show you a slide. When you do nowadays super phylogenetic analysis of the many, many sequences there are, you can actually find organisms where, in contrast to what I told you earlier, there you find C-terminal domains that are active alone without N-terminal domain. And we now have gone ahead to in vitro, by molecular evolution, recreate such enzymes. So this then is a model of the full-length RNA because we were absolutely insistent, and Tateki Suzuki tried everything there is for several years to come up with a structure, because I tell you, the Panzer and the PACE mutations all were in the N-terminal domain. And since nothing was known about the N-terminal domain, we finally had to get to it. So we had a crystal structure that requires zinc in it, which we didn't know before. And then we could do it. So the N-terminus binds here, the C-terminus binds here, and these somewhere, there are another 150 amino acids in there between the N-terminus and C-terminus. So it looks as if the synthetase wraps around the tRNA. It's totally different from any other amino acid synthetase tRNA complex. It has the largest interface area for known synthetase tRNA complexes, and you can see, of course, pylorus does not recognize the anticodon, which is a godsend for all synthetic biologists, because they can then use this for any codon that they would like to do. And this is just because it looks very nice. The wild type structure, and this is a mutant structure, and you see how the tRNA binds. In the mutant, where we get the increased activity and different specificity, opening up the structure decreases the tRNA pile binding and leads to increased non-canonical amino acid recognition, but with lower catalytic activity. How many more minutes? Fine. So a different point is recoding UGA from stop to selenocysteine. Most people do not recognize that selenocysteine, even though it's a genetically encoded amino acid, has no amino acid tRNA synthetase to charge it to tRNA. This is done in an RNA-dependent fashion. Furthermore, incorporation, is not competed for by elongation, also by release factor 1, but by release factor 2. Therefore, a very different model of protein synthesis is happening. And there is cell B, this new elongation factor is required because tRNA-seq is the longest tRNA, has up to 9 base pairs in the acceptor stem. And there's furthermore an RNA element, at least in bacteria, that is immediately adjacent to UGA. And because the RNA element is within the coding region of the protein, of cellular protein, you can't really do any mutagenesis like you can do with other amino acids, because if you move the anticodon, you would also have to move the sequence here. And if you do this, then of course you have obliterated immediately 20 amino acids of your enzyme. So we developed tools that I don't want to describe here to do this and to make any cellular protein that you like in E. coli. But we then did one other experiment. And it was really the absolutely enthusiastic involvement of Marcus, Joan, and Patrick O'Donoghue, where we wanted to work with formate dehydrogenase. And formate dehydrogenase is the only cylinder protein in E. coli